Hello, everyone. Welcome to another episode of This Week in Science podcast, broadcasting live right now. And if you're used to this show, you'll know that right now is the time that we live broadcast. The podcast will be edited, hit all the likes and other things. And I'm being told that there's some kind of a problem to Facebook at the moment. I don't know what's going on. I hope it's going. Anyhow, we're here. We're recording. We're streaming. The podcast will go. And it's time for a show. You ready, Blair?
So ready.
Yeah, yes. Ready as I'll ever.
Ready.
Ever be. Okay. Let's do this thing. Welcome, everyone. All right. Let's start this show. Oops, I hit a button with my finger. Starting the show in 3, 2. This is TWIS. This Week in Science, episode number 935, recorded on Wednesday, July 19th, 2023. Science to sing about. Hey everyone, I'm Dr. Kiki, and tonight on the show, we will fill your head with babies at the opera, scared flies, and missing dark matter. But first, disclaimer, disclaimer, disclaimer. When is a weed a weed? Human necessity and awareness defines it so. As stewards, we look upon the world and let it be, or understanding our impacts, our essential place in ecology, do we take responsibility? What does that mean? How do we define it? Are we a weed ourselves or something part and parcel? Let's consider all possibilities on This Week in Science, coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening, what's happening, what's happening this week in science. What's happening, what's happening, what's happening this week in science.
Good science to you, Dr. Kiki.
And good science to you too, Blair. And everyone out there, welcome to another episode of This Week in Science. We are back again to discuss all the science. Again, without Justin, we hope he's doing well on child duty this week. Hopefully he'll come back sane and happy when he's done.
Probably not well rested, but yes, absolutely.
Absolutely, yeah. But we've got stories about science, as we like to do every Wednesday, 8 p.m. Pacific time. So I have stories about missing dark matter in a galaxy, some issues with private medicine. Did you know babies love opera? And then some other brain stuff. What's in the
I'm
animal
looking forward to
corner?
that. I think that's important to my current interests.
Yeah. It's
I
fine.
brought multilingual science. I also brought some seaweed, some bats, and some scared flies.
I like seaweed, dried and salted. It's very tasty. I don't like, well, maybe scared flies if we can get rid of them. That's the other one.
We need some flies. They're pretty important to the ecosystem.
It's true. It is true. And as we jump into our flying by the seat of the pants science show, no, we've planned it all ahead. It's all very good. We want to let you know that you can subscribe to TWIS, to this podcast, anywhere good podcasts are found. You can also find us broadcasting live every Wednesday, 8 p.m. Pacific time on Facebook, Twitch, and YouTube. And we are TWIS Science on most of the socials out there. And our website is twis.org. If you want to go check out show notes and other things, it's where everything is held archivally, historically. But now it's time for the science. You ready to dive in?
Yeah, what do you got, Kiki?
Okay, let's start with the puzzle of missing dark matter. We've had
That's dark matter's whole thing, is that it's missing.
right. It's one of those things. Where is it? Where's the dark matter? I don't know. Do you know where the dark matter is? I can't see it. Well, we've been trying to figure out where dark matter is in the universe. Our estimates are that it makes up about 4% of the universe, is made up of not visible baryonic matter, or it doesn't reflect. You can't really see it, but it's there. It has a gravitational effect. It has mass. And there is an estimate that most galaxies have about 10 to 70% of their mass, of their content, made up of dark matter. So some researchers at University of La Laguna at the Instituto de Astrofísica de Canarias have discovered that a galaxy called NGC 1277 doesn't have that much dark matter. Maybe at most it has about 4 to 5% dark matter, but they didn't really detect any. So they're looking at this galaxy and they're like, oh, it's an old galaxy. It's what they call a relic galaxy. It's kind of, if we're going to look at galaxies and see how galaxies form, fossils, evolutionary, that kind of thing for the solar system, we look at relic galaxies. So they're like, hey, normal. It's going to be great. This is a prototype. And then it wasn't. And the question is, where did the dark matter go?
Right. So did it start with the right amount and now it doesn't have as much? Or did it never have as much and there's something weird about this that we didn't? Yes. Yeah. Okay.
That's the exact question. So is it, we've seen galaxy clusters in the past where they've blown past each other and gravitationally pulled dark matter away from each other so that the dark matter is missing in one area and it is increased in another area. But this galaxy, we know that it hasn't interacted with other galaxies, at least in a very, very, very long time. So it's kind of by itself. We don't think it's had any interactions that would have pulled the dark matter away. Is it possible that dark matter just kind of leaks away over time?
Huh.
We don't know.
Why? Where? Okay. Where would it go? Why would it do that? That doesn't make any sense.
Yeah. So it doesn't make any sense at all. And these researchers are, they're scratching their heads and they're really wondering how a massive galaxy could form without dark matter, or if it formed with dark matter, where that dark matter would have gone. Is this something that suggests that the standard model of physics is not complete? Or is this something that we just don't know enough yet and we can't answer the question? I mean, really, the answer is we don't know. We know nothing, Jon Snow. So
it sounds like we just picked a bad example to study. Like maybe this galaxy is just weird.
Right, but why is it weird? That's the question we need to answer. Why is it weird?
Well, I mean, before you said what, 10 to 70% of its mass is dark matter? That's a huge range.
Yeah.
So I feel like if you had the kind of like, what was it called? The box plot with the whiskers. If you have that, like there's an error margin. Getting to 4 isn't that far if your range is 10 to 70.
Right. Yeah. So there's maybe some wiggle room, but the question is, why is it down as an outlier from the outliers? So how is it where it is? Why is it where it is? Is it, how did it lose its dark matter? They don't know. So
they just got to study like 1,000 galaxies.
Right. I mean, the thing
and
that
then you
makes
can
me
get
laugh,
a better data set and go, is this one actually weird, or was our estimate of 10 to 70% off?
And yeah, and I mean, that's the thing. It's okay. This is supposed to be a prototype relic galaxy. Prototype. So obviously it's not a prototype. They need to look at other prototype relic galaxies and see if they're consistent with this.
Yes.
Let's do some analysis and see what's going on.
Yeah.
I mean, we're not
is needed.
We're not throwing the standard model out yet, but I love studies like this where it seems really interesting and researchers are scratching their heads. It's good.
Yeah. Well, and you, without being able to go back in time, very difficult to tell if this has changed over time or if it's just, yeah. Mm-hmm.
And like you mentioned at the beginning, is like it's dark matter. We can't see it. We don't really know what dark matter is. We're still like, oh, maybe it's lost in the dust and the gas and the filaments between galaxies, maybe it's just fluffy out there in the universe, or maybe it's stuff that we can't explain yet, and we don't know. So,
Mm-hmm.
dark matter. They have questions about things that are questionable.
Truth.
All right. But these researchers who did this study, of course, they published it in English. They are not American researchers. They are Latin researchers. And so, Blair, you had an interesting food for thought kind of topic to bring up.
So if you and I lived in a country where English was not the primary language, how would we be talking about science? Would we be talking about it in our native language, or would we be broadcasting in English? What would we do? Well, scientific studies are published in English. Mm-hmm. English is the language of science. Might that be a problem? This is a study from the University of Queensland, Australia, from Dr. Tatsuya Amano, looking at the impacts of English being the main language of science, and how specifically the impacts on non-native English speakers on science as a field. Mm-hmm. And so that their kind of disadvantages can range from difficulties in reading and writing papers to reduced participation in international conferences. And so they surveyed 908 environmental scientists from 8 countries with different linguistic and economic backgrounds, and compared the amount of effort required by individuals to conduct a variety of scientific activities in English. And what they revealed, not too surprising, is that there are clear and substantial disadvantages for non-native English speakers. They needed twice as much time to read and write papers and to prepare presentations. Makes sense, right? Mm-hmm. But what I think is really scary is that papers written by non-native English speakers are 2.5 times more likely to be rejected, and 12.5 times more likely to receive a request for revision, simply due to the written English. A lot of them also give up on attending and presenting conferences because they're not confident communicating in English. So,
right.
Again, not surprising. But what can you do with this information? So the bigger picture issue is that these disadvantages are identified, but there's nothing in the scientific community to kind of remove these barriers. Instead, it's up to individuals to figure out how to overcome them on their own. And that is where you can really lose a lot of scientific information, of brilliant minds. And the thing that comes to mind for me immediately is perspectives.
Yeah.
Because if you only have majority English-speaking countries contributing to these roundtable discussions at conferences, reviewing articles, writing articles, doing all these things, do you end up with a kind of homogenized perspective on the science? And do you miss out on kind of different perspectives? Like, how many times have we done stories about, like, oh, before women were publishing studies, this whole area of health wasn't looked at because what about the women? What about all the kind of non-English speaking countries that have different cultural experiences that could inform scientific studies? I think it's pretty important. So they propose some potential solutions in the paper, including supervisors recognizing difficulties faced by students, to journals providing free English editing, which would be pretty cool. And also they suggest funders could offer financial support to efforts working towards overcoming language barriers. So you could maybe get some additional funding to work on your English development. So kind of providing some of those ways to remove barriers and assistance to those to be able to accommodate this universal scientific language of English.
Yeah, it's really still working on this idea that English is our global language, that everybody has to learn English to be able to communicate. And it does leave a barrier to be crossed, certainly. It's interesting about the paper rejection and that the editors don't work more on looking at the statistics or looking at the results of the studies specifically, and then, finding a copy editor or somebody to help brush up the language, or, there's, I don't know, maybe there is something else that, most researchers are working at university institutions. And so maybe there's something that can be done for, this is room for science writing, right? This is a room for technical writers to come in for translation purposes. But
yeah, absolutely. So if there could be more kind of institutionalized assistance, that would be huge. But I think really I want to learn more about this rejection and revision side of things, because this means that process is biased. That's what that means. And if your English isn't exactly right, the science could still be perfect.
Science is never perfect.
Well, yeah, but you know what I mean. It could be sound, right?
Yeah.
It could be statistically significant. You could be drawing logical conclusions. But if your English is not perfect, are you going to get your paper rejected, or are you going to ask for revision? So I think that's the thing that kind of was the spookiest out of all of this for me.
Yeah.
But I also just think it's a nice reminder for everybody. Like, we're first of all very lucky to be speaking the language of science. So I'm thankful for that. Yeah.
Yeah. You think about it though, we've talked before about how science terminology, the actual terms that we use to refer to things, they're usually based in Latin. They're usually very specific roots, which, are still Western, Romance kind of based. But at the same time, science itself is hard to get into because you have to learn how to speak science as a language, so, it's maybe easier for people who have a background in English speaking, but I don't know. It's a very interesting question all around. We shouldn't have barriers. It should happen, but how do we make it possible for people to communicate better? Is this where AI comes into play?
That's interesting.
Babblefish? I don't know.
Yeah, Google Translate just
you
just write my paper in my native tongue and just have a very garbled translation just delivered to the journal.
Yeah.
I don't know. That's a really good question, especially with like the it's not called Google Glass. What is it? The one that Apple made. Oculus? I don't know. The goggles, the face goggles, the AI face goggles. You can
computeronmyface.com.
Yeah. Yes, exactly that. You can potentially wear that in the future and read text in front of you in whatever language you want.
Right.
So if that's the case, does that make things more accessible and more universal?
Yeah. Right now, it's interesting that AI is often identified as being written by, like, papers that are being written by AI are identified as being written by people who don't speak English as a native tongue in studies recently. So I think that's another interesting point to it. But I don't know. So many interesting levels.
I don't know. But to your point, if we get our universal translator from Star Trek, none of this matters. Yes. Just speak whatever you want.
Say what you want. The computer will fix it. Computer, engage in communication. One thing that's important to communication is figuring out how to get people to seek out healthcare when they need it, how to get people understanding what insurance benefits they have. Like, our system here in the United States is
made to be confusing.
Yeah. It's kind of made to help people fail.
I'm familiar.
There is a study out this last week from the British Medical Journal, BMJ, and it is in relation to private equity firms that have been investing in and basically becoming owners of healthcare companies. So nonprofits or other hospitals or insurance companies are being purchased by private equity because it's a great investment, because healthcare, there's so much money in it right now. Oh my goodness. But there hasn't been a lot of speculation, or there is speculation as to its impact, but there hasn't been any research on it to date. And so this study did a review of 55 other studies, 47 in the US, from the last couple of decades. And they found that nursing homes were the most commonly studied settings than hospitals and dermatology facilities. And all sorts of quality in the studies, all sorts of methods, all sorts of weird things. So they finally ended up like putting it all together, and it's a mixed bag, really. So private equity, for a large part, though, for 9 of 12 studies, they had higher costs to patients or payers at health facilities once they became owned by private equity firms. 3 found no differences. None showed lower cost. There's 27 studies that looked at healthcare quality, and 12 of them found that healthcare quality went down, and it was actually harmful to the patients. 3 studies were beneficial. 9 were kind of mixed, where it was like, some things are good, some things are worse. You're probably just, dealing with efficiency and getting some things, certain outcomes. When they looked at nursing homes separately, there was a mixed impact, but overall there was a degradation as opposed to an improvement in quality of care at nursing homes. So fewer staff, less skill, all sorts of things. The result, really, they note, is the results of this study confirm the need for increased rigorous research on private equity ownership in healthcare, particularly its impacts on health outcomes and system costs, and in other non-US settings, such as Europe.
So what you're saying is, when you're worried about the bottom line, the quality suffers.
Yeah.
Where have I seen this before? Oh, right.
Yes.
Capitalism. All of capitalism.
But it's one thing to just like have our anecdotes and be like, whoa, American healthcare, we pay more money, and whoa,
for less.
Healthcare outcomes are weird and people are in debt. And it's like we kind of have all this stuff put together, but then when there's an actual, review of research that's done to say, here's the data, and more often than not, when healthcare is privatized, when it becomes for profit, there is a deleterious impact, or it doesn't get better. At least it's not better for patients. Yeah.
Well, you want to spend less money and make more money. How do you do that?
Right.
You cut corners and you cut care.
I mean, it makes sense, right?
Yeah. Absolutely. And it's another reminder of why that shouldn't be how this works.
Yeah. I feel like this is the kind of study that Justin would have brought to talk about on the show, but I do think it's important to, when there are studies like this, analyses like this, to kind of highlight them, and we can add them to that corpus of information that we have when we look at the world, right? And we look at it and go, okay, what's happening? Who am I voting for legislatively? Are there regulations that can, are there legislators who are looking at this? Can I write a letter to my representative? What can we do?
Yeah. It's a great question. In America, I don't know. It's kind of baked into the system. So pretty tough, especially because the more kind of radical stuff that they were talking about doing with healthcare here, you were still going to be able to have your private option. So that doesn't really get rid of this issue. But hey, if somebody comes out there and says that they want to put caps on things and basic standards of care and all these sorts of things, and pay closer attention to insurance companies, then yes, then you vote for that person. But I'm not going to hold my breath, meanwhile, mainly because I won't be able to pay the medical bills when they show up to take care of me, because I passed out.
I hope you didn't pass out. Don't pass out, Blair.
No.
Don't pass out. Oh my goodness. Let's talk about sex.
Oh, yeah.
And seaweed.
Yeah, seaweed sex. Did you know that seaweeds have sex?
No, wouldn't think of it that way. Nope.
Reminder that seaweed is not a plant. It is not in kingdom Plantae. It's a
Oh, what?
protista. It's an algae.
Oh,
That's right. Yes.
that's
And so
right.
they have sex. They have sperm and egg. It's not exactly like animals, but it's more similar to how animals mate than to how plants do. That being said, plants on land often use pollinators. And we talk about them all the time because it's like our entire lives depend on pollinators and pollinating plants because we eat those. But they can do that by bees brushing up against, getting covered in pollen, or there can be seed pods that travel on the fur of animals, or sometimes they're like helicopter shaped and they fly, or sometimes seeds can pass through the digestive tracts of animals and then actually germinate via feces. But so on land, we are very well versed in how pollinators work in concert with plants to kind of this mutualistic relationship. The pollinators get something out of it and the plants get to continue on their family line. Well, it turns out that seaweed, they might have a similar arrangement going on, which is brand new information because up to this point, the expectation was that underwater, the movement of currents and buoyancy disperse and fertilize seaweeds.
Mm-hmm.
They're broadcast spawners. That's it. They just release a bunch of sperm. It gets carried along the waves. They somehow find eggs in that giant ocean and they get fertilized. And yeah, it's great. It's like a needle in a haystack, but it works every time. Well, new research says there might be pollinators at work underwater, which is pretty cool. So they found some examples of mutualism among seaweeds and animals. This is researchers from the Sorbonne University. They found isopods, which are these tiny little marine invertebrates, 2 to 4 centimeters long. They look kind of like albino beetle-y things with too many legs. They can increase fertilization success in red seaweed, Gracilaria. They feed on this small algae that grows on the red seaweed, and then the seaweed sperm can attach to their bodies, and they carry the sperm just like a bumblebee carries pollen to female seaweeds, which helps them fertilize exposed eggs. And in return, they get a meal, and they also get protection by hanging out on the seaweed. So based on that discovery, they're like, oh my God, what else is happening underwater? And so they looked further and they found more examples of seaweeds having sex, basically having their fertilization occur due to animals helping them out. And so they actually looked at animals that previously were blamed for seaweed problems, grazers, to see how they might hurt or help these individual seaweeds. And so they looked at urchins, which if you are familiar with the story of sea otters, especially in the San Francisco Bay Area, they always talk about how, the sea otters were trapped and furred. Urchins exploded in population. They overate the kelp, and then the kelp forests were kind of destroyed. Sea otters are coming back. They're controlling the urchins. The kelp forests are returning. It's really cool. So this is this whole idea that like urchins are hurting more than they're helping the seaweeds, these kelp, right? And so they wanted to look specifically at urchins because of that, because that's the whole narrative we get. The urchins are hurting the kelp.
Are they just the bad guys? Yeah.
Right. And they found that gametophytes, so sperm or egg, are surviving through the urchin intestinal tracts. They were able to keep gametophytes for a few weeks that had been expressed through feces, and noticed baby kelps growing from the feces of both of these urchin species. And none of the gametophytes kept outside of the feces were fertilized. So being eaten by the animals actually helped them gain fertilization. They're not sure how. Their guess is that it has to do with proximity. So if the urchins are munching off this plant and munching off this plant, then they might have sperm and egg in their intestines. They meet, and then they're able to fertilize. By the time they're pooped out, They're a little baby kelp.
Oh my God. So where did you meet? Oh, we met in the
Oh.
intestine of a sea
Yeah.
urchin.
Oh, it was magical.
Love at first
Um,
sight. Or
yeah.
bite.
The other guess is that it could have to do with some sort of chemical cue in the feces, but more research is needed. But so essentially, they found repeated examples of animal-mediated fertilization in the ocean, which means a couple things. First of all, seaweeds evolutionarily are a lot older than terrestrial plants.
Right.
So it's the age-old question: did this evolve multiple times? Or did animal-mediated fertilization first show up underwater? Either way, very cool twist on how we think pollination started. Because the expectation was that it was very much a terrestrial process. And so that is not the case. So either terrestrial plants popped onto land already kind of with this mechanism at play, or it's convergent. So
Huh.
my
I
money's
mean,
on convergent, but whatever.
Yeah, I mean, perhaps I would guess convergent. But,
Yeah, there's just too many factors. Like,
there's factors.
there's no bumblebees in the ocean. So how does this relationship permeate onto land through multiple different species, different kingdoms, like different everything?
Mm-hmm.
How does it permeate through? Like, it seems harder than to just stumble across this mutualistic relationship again. That seems much more likely to me, but
Right. If you're thinking the parsimony of it, then yeah. And I'm going to say that reproduction is probably a lot more opportunistic than it is deterministic in that sense. Yeah.
Yes, absolutely. Yeah, so there you go. Next time you're looking at some kelp at the aquarium, think about them using those urchins or those isopods.
Well, I think this
baby
is
kelp.
Little babies pooped out of urchin guts. But I think the really interesting aspect here also is the change from urchins are bad, okay, to realizing that urchins are part of the ecosystem, and that urchins, yes, when the sea otters, their predators went away, they went out of control. And that's what led to that decline of the forests. But they live in the forests, and they are an integral part of what happens there. So this just, I don't know, it gives more information about the nuance of those relationships.
Yeah, absolutely. And if urchins have been around for millions of years and kelps have been around for many, many millions of years, like
many long times,
why would a deleterious relationship continue? It doesn't really make sense. It's it makes a lot more sense that they would evolve side by side and kind of find this mutualistic relationship. It's not always the case, but especially with the kind of they've been buddies for millions of years.
Yeah, kind of sort of buddies. I eat you. Maybe we can make this work for both of us. Okay, this is good.
I just gave you a little haircut. No big deal.
I love it. I love it. There's better uses for that ravenous appetite of the urchin. Yeah. So Blair, are you going to take your baby to the opera ever?
To the opera? Not right away.
To the opera.
I think I should wait till he's at least a couple months old, right?
Maybe once the baby can actually see things from a distance. Make sense of movements and everything. Well, anyway,
not poop every 30 minutes. Yeah,
that's a big one. Maybe not. Have a tantrum every. Okay. Researchers at the University of Toronto Scarborough have just published a study in which they took a bunch of babies to the opera, and they took the babies to the opera and looked at how the babies behaved. So where their attention was oriented, how long their attention was oriented there, how they, where they were looking, how they were looking there, and what they saw from examining 120 babies from 6 to 14 months. So still pretty young. They had 61 of those babies watching the concert in person. 59 of them watched it on Zoom. So they watched a recorded version of the concert, and they tracked with heart monitors, and they determined when the babies were looking at the stage or looking away and all this stuff. And they found that all the babies really liked the opera. They all paid attention to it. They all were like, oh, and kept having attentional bursts. However, the babies that watched the opera over Zoom were more likely to get distracted. They're at home, they're comfortable or whatever. So the babies were more likely to break their attention more quickly than the babies at the opera. So the babies that were actually at the opera were more likely to have their attention held by the activity on the stage. What they think is interesting about this is that it suggests that not just viewing that kind of enriching content, so Teletubbies or whatever else on a screen, that there's something about the live performance, there's something around being other people in a social environment that led to the quality of the attention that the babies were able to give to the people on stage, that they were enjoying the sound and maybe the energy of other attendees. We don't know that per se, but the attention live and in concert was stronger.
I love this. I think the first thing I thought of about opera actually is that there's similarities between opera and what is called parent speech, which is when you have your elongated vowels and you have your high highs and your low lows in your voice. It's different from baby talk. Baby talk is like nonsensy language, right? But parent speech is when you kind of have that sing-songy voice and you go, oh, hello, how are you doing today? And that helps them develop language, and it's interesting. And opera's kind of like that. Elongated sounds, high highs, low lows. It could pique interest. And so I could kind of see that, although, a lot of opera's not in English. So are these English speaking babies? Not that they speak yet, but are they from English speaking countries? And what language was the opera in?
This is McMaster University. It's Canada, Toronto, Canada.
Sure. Yeah.
Yeah, and it wasn't a full-length opera. This is like a baby version opera.
Yes, I would imagine that'd be
12-minute show. We're not talking 3 hours at the opera for the babies. Yeah, so, and it didn't depend on what they could see, whether or not the babies had any previous concert or music engagement, that all the babies, regardless, were engaged by it. But there's something about listening and experiencing with others that makes the difference. And so they ask the question, if a baby is frequently brought to these kinds of events, is it going to shape their foundation for engaging in music in the community later in childhood? So the question, if they were going to do long-term study, is, do babies that are taken to more of these things engage more with music generally? Does it shape lifetime habits of interacting and engaging?
I'm also curious if they took babies to the movie theater to watch an opera. Would it be
that's right? A great control.
Yes, because you have your screen, but you also have the social interaction of the other people in the theater with you. Like, what part of it is what they're responding to? Is it the actual physical actors on the stage being in the same space with them? Or is it, as you're talking about, this kind of social experience where you're in a room paying attention to a singular thing amongst others?
They need to do more work.
Yeah, it's very cool. I mean, I love it. It also maybe is a good reminder of where babies born during the pandemic maybe had some trouble
Yeah.
and are continuing to have some trouble, because if they only interacted with people besides their parents through FaceTime or through Zoom, is this related? Is this something where they, their attention span or their interest in people or any of these sorts of things could be impacted by the fact
Yep.
that they weren't in a social space, which isn't surprising, but is still important for us to look at for sure.
Yeah. And that's it's like kind of a natural experiment. We've got pandemic babies and post-pandemic babies. So
Yeah.
that's going to be interesting research directions for sure.
Oh, we're gonna be studying that for another 20 years at least.
Yeah. So Blair, take your baby to an opera or
I'd love to.
a live concert in a park.
Sesame Street on Ice. You got it.
You got it. Yeah. Teletubbies live and in person.
Oh boy, that would be wild. Do kids even still watch Teletubbies? I feel like that's
No
old hat
idea.
at this point, right?
I'm too old. I hope they're old hat. So we've gotten rid of our COVID segment of the show, but I wanted to bring a COVID story this week because I think it is actually very interesting and related to some questions that we had a lot during the pandemic. Specifically, so many people were asymptomatic and still are asymptomatic for COVID-19. Catch the virus, no symptoms whatsoever, and then you test positive, right? So why is that? Why is it that some people are more likely to have symptoms than others? And this research study used a COVID-19 smartphone app that was developed by COVID-19 Citizen Science Study. And they took advantage of genetic data stored in the U.S. Bone Marrow Donor Registry, Be the Match. And they were able to collect DNA, have these DNA samples, and to correlate COVID-19 experience with individuals in this, and analyze genetics of about 30,000 people without having to go out and do cheek swabs or anything like that, because all of the information was already there. They found that 1,428 unvaccinated individuals had a positive COVID-19 test. 136 reported no COVID symptoms whatsoever. And then they determined, excuse me, I'm going to sneeze, I think. It's itchy nose. Okay. They found that there was a gene variant of a human leukocyte antigen, or what's called an HLA gene, in individuals with asymptomatic infection. The gene is HLA-A-B little star thing 15:01, whatever. It's a
little star thing.
Little star thing. So this is a little mutation in the human leukocyte antigen, which is part of the immune system and encodes for particles that are T cells, or that T cells recognize. They are important in the immune response to pathogens, and they vary a lot. Different HLA for different people. This particular variant, according to the researchers, their article on The Conversation, present in about 10% of European ancestry individuals. And if you had the variant, you are twice as likely as other people to be asymptomatic if you got infected. And 2 copies, if you had 2 copies, one on each of your chromosomes, 8 times as likely to be asymptomatic. So it's not necessarily, people like, there's genes in nature, right? Nature, nurture, everything. But there are genetic differences between individuals. And this is not only going to be important for COVID-19 and how we move forward, but understanding individual responses to infection in other viruses in the future.
Yeah. So, for example, if you could figure this out ahead of time, if you could figure it out earlier in the pandemic, then you could have people with these predispositions test every week.
Right. Yeah. You
No
are more
matter
likely
how
not
you
to
feel.
show. Yeah. You
Yes.
feel great, but you might have, yeah, exactly. Yeah. You might be a carrier.
And that's going to be a problem because like HIPAA and all this other stuff. And you're not going to want to have to disclose to your workplace your genetic makeup and all these sorts of things. But if you have this predisposition and your doctor says, you should really be testing weekly for COVID, and you're a good person, you're going to do that. And then that can help prevent spread, because that was a huge issue with like the second, third, fourth, however many waves that we had of COVID. The first waves were based on just not knowing anything about the virus, but later on it continued. We got these bigger waves because people didn't know they were sick.
Yeah.
And so, it would be so nice to be able to identify, okay, I'm one of those people. I need to be testing myself regularly so I don't accidentally get people sick.
Right. I might feel fine, but gotta make sure. Yeah. It's interesting though, these individuals, the HLA gene, their mutation did make them more effective at fighting off the virus. So that's really, it just keeps the viral particle number low. You get infected, the virus gets in, it starts to get in there. If you have memory cells that are like that, because of your genome, are like, I've seen this before, attack, you attack, and they attack more effectively. They can, tamp down the infection more quickly, so you don't have those immune reactions and you don't feel bad, but there still might be virus that you are releasing.
Ooh,
anyway,
the other thing you could do, edit everybody's genes. What could go wrong?
Genetic
Just
modification.
CRISPR the whole population. Just change everybody's genes so that we all have that predisposition and none of us will get sick again.
Okay, Blair.
Just kidding.
I see you.
I'm just kidding. Never mind.
Oh, yeah. I mean, that's the question, right? This is diversity among humans, and that's part of the thing that allows people to survive well, because there's a lot of genetic diversity, right? Some people do well, others don't, depending on what we're fighting off. So you can't just make everybody the same, because then if something shows up that we're not ready for, what do we do then?
When something shows up?
When. Yes, exactly. I hope we just showed up on your radar. This is This Week in Science. Thank you so much for joining us for another episode of This Week in Science. We have a second half of the show coming up here very soon. And before we jump there, I would love to remind you that TWIS is listener supported. And one thing you can do is just tell other people about TWIS. Spread the word, help share the science, and get more people watching or listening to TWIS, and that'll help so much. The other is, if you really, really enjoy TWIS, become a Patreon supporter. Head over to twis.org and click on the Patreon link. That'll take you to the Patreon page where you can choose your level of support. Anything helps. We really, really can't do this without you. Thank you for your support. All right, coming on back for more This Week in Science. It's now time for that part of the show that everybody loves, full of animals. It's Blair's Animal Corner with Blair.
She loves our creatures, great and small. Biped, millipede, no pet at all. You want to hear about animals? She's your girl. Except for giant pandas and squirrels.
What you got, Blair?
I have bad news for bats when it comes to loud music festivals. Kiki, you like to go to music festivals?
I do.
Yeah? You think bats like it when you go to musical festivals?
Nope. Probably not. But I never really thought about it.
No.
Thanks.
And that's kind of why I wanted to bring this story tonight. I didn't really think about it either. University of West of England is presenting this week the first evidence of negative impacts of music festivals on bat activity, which, that makes sense. They found that even in the absence of additional anthropogenic factors, commonly associated with festivals like lighting or habitat disturbance, loud music alone is enough to disturb several bat species. They observed declines in nightly bat activity along woodland edges of around 50%. It was 47% in Nyctalis epicticus species. And then species that are usually super tolerant of humans also decreased their activity. This was P. pipistrellus by 32% during music playback periods. So the way they did this, they looked at 10 sites in Southwest England and South Wales between August and September 2021. They found dark semi-natural landscapes along woodland edges adjacent to grasslands or pasture habitats. And so they resemble conditions that are usually the case for UK music festivals. And then over the course of 2 nights, the first was a quiet control night, but the second was a night with loud music playback. The loud music was at volumes reaching 100 decibels, which is comparable to noise levels at UK music festivals. They played 5 common songs, each representing a different music genre. I definitely want this playlist. I have no idea. And featuring different tempos and frequencies. They stitched all of them together to produce a 10-minute medley. And then they watched what the bats did. For 2 hours after sunrise, noise alternated between the music medley and then 10 minutes of ambient background noise. And they recorded about 2 meters from the speakers, and then 20 meters, and then 40 meters to study noise disturbance impacts on bats at these different ranges. And so they found that playing all of this music had an impact on these bats, the 47% or the 32% that I mentioned before. And so the hope is that, first of all, they can expand the experiment to include a range of different habits and locations. So there might be additional environmental elements at play so that they can find areas where bats are more tolerant. Maybe you can have your concerts there instead. But also they're hoping that this will impact policies related to noise levels for these festivals. Now, the thing that I wanted to throw out is, yeah, bats don't like loud noise. Lots of animals don't like loud noise, especially at night.
Right.
But by them kind of isolating their variable of loud noise, I actually think they made the impact of the study worse. And let me explain. You set up a festival.
Mm-hmm.
You have lights. You have people.
Yes.
You have all these things. That could clear bats out of the area for a while. And so
Right.
that could actually remove bats from the situation. They could go to a different area and have their habits not impacted during the festival because they cleared out.
Yeah.
Whereas if you just are like, you're sneaking up on them, and then bam, you turn on the music, of course that's going to impact their behavior.
So this is more like researchers scaring bats away. Just scaring them, like, what's going on? I gotta run. Yeah.
Which, the way these music festivals work, it takes minimum a week, right, to set up cyclone fencing, parking areas, booths, like all this kind of stuff. They do sound checks, they do all these
lights,
things.
Everything.
Kind of warn wildlife that something's happening. And whereas that still is probably not ideal, that you're clearing wildlife out of an area that is a wildlife area. Humans exist and we do things in nature. So you're not going to stop that. But that's why I feel like they need to redo this study with all of the things. Like, really, they need to just study bats at a music festival.
Right.
To see how they behave.
Well, what I wonder also, insects are going to be change their behavior as well. The insects in the area are not going to want to be trampled in the ground. They're going to either stay away from the loud noises themselves, or maybe the nocturnal ones be attracted to the lights. But
yes,
perhaps and maybe there is something related to where the insects go. Do they go to the camping area? Do they go to the parking area?
Right.
Do the bats move related to that? What are the specifics? I mean, what is the goal of this study? Tell people not to do music concerts because of the
Right.
bats? Or what are we
Right.
trying to do here?
Right. No, that's a great question. And I think that's part of why they started doing the study where they were kind of checking at different distances. So you could figure out, okay, we know there's bats in this habitat. You have to be 40 meters away or all the speakers have to be 40 meters away from the tree line or something like that. That would be helpful. That would be super helpful information to have. But again, to your point, does it not matter if they then post up a bunch of lights that attract insects and then the bats come to where the lights are anyway? Then your 40 meters doesn't matter.
Mhm.
So I feel like this is one of those situations where they tried to decouple variables, but the variable is music concert. It is not
Just the
decibels.
sound.
Yeah. The variable is presence of a festival, no presence of a festival.
I mean, yes, we know that sound impacts other sonar using animals in the water.
Mhm.
So dolphins, porpoises, whales are impacted by loud sounds underwater. And so this, kind of follows on the heels of how animals who use these senses for their existence in an environment could be impacted. I mean, I run away from really loud sounds myself sometimes. But yeah.
So I don't know. Go to your concert is my point.
Go to your concert. If you see
Yeah.
a bat, enjoy the bat.
Yeah. Unless you think bats are scary, then you might avert your eyes.
Would you?
You might,
yeah.
especially if you were a fruit fly. So my other story for tonight is all about the what's going on in our brains when we avert our eyes from a scary or disturbing image. This is something that appears to be conserved all the way from fruit flies. And this University of Tokyo study found the specific cluster of neurons that is responsible for this behavior. These neurons release a chemical called tachykinin, which appears to control the fly's movement to avoid facing a potential threat. And they do think that this is conserved, and this is why we do this as well, why we will avert our eyes from something scary or disturbing. It's a single cluster of about 20 to 30 neurons, and it regulates vision when in a state of fear. The way they figured this out, I actually really like this experiment design. So they used puffs of air to simulate a physical threat and found that the fly's walking speed increased after being puffed at. So this is the first step. They're training flies on how to behave around a threat, basically. So they need to make sure that when they're testing imagery, that it is associated with a threat. So because you don't know like what's going on in a fly's brain and what is scary to them. So you have to couple it with some sort of stimulus to indicate that threat. And so they would choose the puff-free route if offered.
Otherwise,
and so that proved
puff air.
that proved that indeed the puff of air was a threat to them and that they preferred to avoid it. So then they placed a small black object, roughly the size of a spider, 60 degrees to the right or to the left of a fly. And on its own, the object didn't change their behavior, but when placed following puffs of air, the flies avoided looking at the object and moved so that it was positioned behind them. They didn't want to see it. And so
if I don't look at it, it's not there.
Exactly. Yes. So then they suspected this neuron situation. So they mutated flies where they altered the activity of certain neurons so they could hone in on what neurons are responsible for this thing. And so when they mutated flies, they kept their visual and motor functions. They would still avoid the air puffs, but they did not respond in the same fearful manner to visually avoid the object. So basically, they still didn't like the air puffs, but they didn't care about the object at all. So because they associated it with this very specific group of neurons, they were able to figure out this is what's causing it. So next, they want to figure out how these neurons fit into the broader circuitry of the brain. They know it's in this visual area of the brain, but they don't know kind of how it fits in. And so they want to see from where the neurons are receiving inputs and to where they are transmitting them to regulate their visual escape from objects perceived as dangerous. And so they want to be able to draw a complete circuit diagram of how fear regulates vision, which, aside from just being really interesting and helping us inform how animals behave in certain situations, if it is something indeed that has been conserved into humans, this could help with the treatment of psychiatric disorders stemming from exaggerated fear, like anxiety or phobias. And so there is potentially an application to this in the end that could help with therapy in humans.
So this is fascinating because there are a number of therapies that involve stimuli that you kind of go, how does that even work? EMDR is, I think it's EMDR is a therapy where it's eye movement.
Desensitization and reprocessing therapy. I did not know that. I Googled it.
Yes, thank you. That's what it is. But it's you revisit stressful experiences from the past, and there are eye movements that are involved in going back and thinking about them different ways. And there's been a lot of success for people in using this EMDR therapy to help them overcome traumatic past experiences and reduce anxiety and depression. I find that fascinating, just number one on its own, but then to now be able to say, there's these potential circuits involved in a fear response for the eyes, for vision and what's happening in the brain, that maybe there is a much deeper link that explains it in some way. But fruit flies, what? Fruit.
They avert their eyes from something scary. And someone in the chat room, Potato Dust, wants to know, could it also be a physical response given eyesight is a significant anatomical feature for the flies, alternative to optical? So that's a great question. The way they kind of selected out for that was the secondary part where they had the mutation where they didn't have this neuronal response and they acted differently. They didn't reorient their body away from a puff of air like they would if it was purely physical. They were specifically walking in a different way where they didn't mind if they could see the object. So really good question. And this study design was very specific to try to make sure that this is what was going on.
It's fascinating. I am fascinated by, I guess it would be orienting the body to enable escape, to enable flying away. But I mean, what good is in humans, avert your eyes, cover your eyes. We watch a scary movie, cover our eyes. But that, is it like object impermanence? If I can't see it doesn't exist. Like,
that's
it's
what
gone.
I think. Yeah, that's absolutely what I think. I think it's a desire to not know.
I cover my head with my blankets at night when it's dark and the scary sounds come from the closet.
Yes, absolutely. That's, I mean, whenever we're talking about animal intelligence, we always associate animal intelligence with like a 3-year-old human or a 5-year-old human. That's when those impulses are still extremely strong. If I don't see it, it's not real. So for that to kind of originate in fruit flies would not be surprising at all.
Yeah, it just really does.
To your point, like, evolutionarily, why? Because if a threat is there, it's there. It's gonna eat you, regardless of whether you see it or not. So is there something with like stress hormones where if you don't see it, then you don't get your stress hormones elevated, and then I don't know. I wasn't even thinking about that. Why would they do this evolutionarily? There has to be a reason.
Yeah,
Huh.
I mean, there's fight, flight, or freeze, right? We used to talk
Right.
always just about fight or flight, but freeze is also a reaction for many animals. The deer caught in the headlights, or a rabbit, or, you know, so it is something
Yeah.
that happens. But yeah,
Right. So
maybe
is it
res-
avoiding the freeze response? Is that the idea? Is that like if you got overloaded with stress, would you freeze? And so avoiding the visual contact, does that allow you to escape?
right. Can I get away now?
Interesting.
Yeah,
Interesting. I like it.
I'm speculating.
More study is needed. University of Tokyo, you listening?
I want to
Hopefully
know more about these.
everyone's very fluent in English based on our previous conversation that we had, and they could listen to this podcast and follow up with their fruit fly research. Thank you so much.
I'm sure that they are listening to this podcast.
Yeah, oh,
Absolutely.
for sure. Yeah, absolutely.
Yeah. And then, yeah, I guess the next question would be cortisol levels, stress hormones. Look at all the ways that those can be manipulated as well with
Yep.
the visual response versus just the puff of air response versus, yeah. How does that all change?
Yep.
I don't know. I'm going to follow right on with your Animal Corner with a study that was published a week ago in Science Advances. Researchers published their work looking at little tiny primate animals that they're tiny wild
You talking
gray
about
mouse.
marmosets?
No, wild gray mouse lemurs.
Oh, tiny
Yes.
primate, I think marmoset, but you're right. Mouse lemurs are teeny tiny.
Little teeny tiny mouse lemurs. And so they can live in a lab. They live about 3 years. They also have differences in individual fitness and body shape, size, etc. Researchers were looking at the big question of how does cognitive performance influence fitness in the wild? So, cognitive fitness being like, how well are you exploratory? Are you checking out novel things? Do you like that? Are you problem solver? Are what aspects do you remember things? You're learning and memory really great. How does that impact your ability to survive? Fitness, as we define it in natural selection and study of evolution, excuse me, is related also to individuals' ability to pass on genes to a next generation, which then also survives. So it's like this heritability factor is fitness. But in this study, they were trapping wild gray mouse lemurs, and so they couldn't track the reproductive ability and the reproductive success of these 198 animals that they were able to trap and put through various different cognitive tasks to see how their personalities turned out. So the researchers had these little gray mouse lemurs go through a number of different cognitive tasks: a puzzle box, which is problem solving, can you find the food? A maze, which is spatial memory. Open field exploration, just a big open space, kind of testing it out and seeing what's there. A cylinder task, which is related to inhibitory control. So, how much can you, like the marshmallow test, can you not do something? String pulling, which is causal understanding of if you pull on the string, something else happens. Or then neophilia versus neophobia, which would be a novel object. The issue I see with this study is that they couldn't test all of the animals on all of the tests, and they kind of mixed and matched, and they were like, well, some of the animals were in this test and others were in that. And so we got this general idea of personalities of the animals that we were then able to link generally to their fitness. But instead of actually testing their fitness, they related it to body size. So if you're old and big, they were like, great, oh, you're super successful, which is weird. So a lot of the details of this study I have questions with, but they correlated it with all of these individuals and determined that exploratory nature is something that was related to fitness or becoming old and big. So those that were old and big were more likely to be exploratory as well. They didn't know which individuals were going to die or how many kids they had had, but they determined that if you're older, you're more likely to have had more kids.
Sure.
And so then you're more fit because you're older. So there's so many assumptions to this study that they bring up. But their bottom
aren't
line...
you more likely... Sorry, go ahead. The
Yeah,
bottom line.
I was just going to say the bottom line to their study is also kind of a well-dubbed bottom line, which is that there are cognitive determinants to fitness and survival.
No, absolutely. But the exploratory nature one really gets me because, of course, you're going to be more exploratory if you're older because you have experienced more. And that's we know that already with animals across the line. The longer you live, the more familiar you get with different scenarios and the more comfortable you get with new things. That's very normal.
Yeah.
That's we already know that causal relationship. So that's not helpful.
Yeah.
But yeah, that's if you can't test every mouse lemur on every study, why do you have so many?
Yeah, they went. Which I find, I feel like they went out and did a bunch of. They were doing different studies, and then they lumped a whole bunch of things together for one study. So I'm wondering if this is like a post hoc kind of p hacking. I don't know. If like, oh, we have all this data.
Absolutely.
Now let's ask the question.
Oh, I love that. Yeah, you're right. That really seems like, a bunch of my friends were studying on mouse lemurs and had these different experiments. I wonder if I forgot to do a doctorate. Let me just, can I borrow all your data? I'm sorry. I don't want to insult the person that wrote this. That's mean. But no, I agree with you. It seems very strange that you would have, you're always trying to isolate variables. Why would you add 6 different tests? That's wild.
And especially like if you're not having all the individuals doing all the tests, you're not really able to determine overall personality. Like this one individual or this group of this is going to act this way, have these personality types. This is the way that it works. And so, they're lumping them together. I guess what they really want to get at is that, it's not only being the older and exploratory, but there were other successful strategies. And so there are multiple ways to be successful. They couldn't really pin down a particular intelligence quotient to be able to say this is it about intelligence, right. Which, as we know, intelligence is really hard to nail down in the first place. And
there's different kinds of intelligence. Yeah.
Okay. Yeah. So, okay. Cognitive performance generally is heritable and could influence fitness. And, down the line.
That sounds correct. Yep.
Yeah. So anyway, it's one of those studies. I just thought it would be fun to bring it to hear your...
Yeah. I also just like picturing those mouse lemurs doing those experiments. That is also very enjoyable for me. And I will continue thinking about that for a few days, I think. So I appreciate that as well.
Let's think about mouse lemurs now. Doing
Pulling
little
levers.
tests.
I love
Pulling
it.
a string. What's at the end of this? Oh my goodness. What am I doing? Researchers, speaking of animals and other things, we do a lot of research that involves animal cells, and it's always kind of a goal to remove animal cultures and other influences from research that involves human cells. Researchers from the University of Michigan have published in the Annals of Clinical and Translational Neurology their work that gets rid of mouse matrices to support brain organoids. So what do I mean when I say mouse matrices? Well, when you have a bunch of cells, they kind of have to be supported by something. And so there's an extracellular matrix that often has like fibrinogen, support collagen, supports proteins and compounds in it that helps the cells grow into a structure. And
Is that like bleached out animal cells, basically? Or
sometimes yes, sometimes no. It depends
Okay.
on what they're doing. And so for what they're talking about with brain organoids and these mini brains, they hadn't been able to support all of the human cells that have been used in these human brain organoids with a human extracellular matrix. They haven't figured that out yet. And so they've been using a substance that has come from mouse sarcomas. So from cancerous tumors in mice, it's called matrigel. And that's what's been used by everyone for these human mini brains, which we use to study disease and development of things in humans, right? We want to know what's happening, but we're using this animal tumor tissue. So how is that impacting the results? Might not be the system that is the best system. So they created a system without any animal parts in it, and they are able to use it for the neurogenesis of brain organoids. It's very exciting, and they have developed it from something that is called fibronectin, which is a protein that supports stem cells in human tissues and supports them as they mature. So fibronectin, and they've also added a polymer as well. So it's not just animals, but it's also a porous polymer that's a scaffold. So that polymer could probably be 3D printed or grown if it self-organizes. But the thing about this study that I love the best is like they're working on this. They're trying to do it, and then COVID-19 hit, and nobody was able to go to the lab for like a month. And their cells just kept culturing for that month without them, and they did really well with this particular mix. So that's kind of like, oh, hey, we couldn't come and help it, but look they grew really well.
Gosh, I can't imagine setting up an experiment like that and then not being able to go in, and just for a month it being Schrodinger's science experiment, and you're just like, either everything's dead or it worked.
Yep.
Wow.
But this worked really well, and they determined that the organoids developed like a cerebrospinal fluid, which is the normal stuff that flows around our brains and helps with getting nutrients around and also with removing waste products. And so this is really great because that's good, and it means it's developing normally. So yeah, leave this alone for a month in the lab. We've got a little tiny little brain system, all human, no mouse. No mouse at all. And my final study for the night is a story about how researchers were looking at the internal clocks in our brains. So we know we have an internal clock that is influenced by sunlight. It is influenced by our internal systems as well. Some people are genetically more morning larks. Some are more night creatures. But our brains work, and then there's a trickle-down effect in other organs throughout the body that also have clocks, and so that main clock impacts all the other clocks, and researchers were like, okay, let's look at this, but we also want to know not just how that clock works, but how we perceive time and what's going on in the cells and in the brain as we're perceiving time, whether it's longer or shorter. And of course, to study how humans perceive time, these researchers looked at mice. And in this particular study, they took what's a pretty cool use of optogenetics. But you think optogenetics, it's light and genetics where it's involved. But what they did is they used the optogenetics to influence an area of the brain deep in the striatum of the mice, and they increased or decreased the temperature of the brain. And so instead of turning on light and stimulating a gene and making neurons active, they heated up or cooled down that part of the brain. And they saw that when they heated it up or cooled it down, it changed the activity of molecules, not just the neurons, but like molecularly, the speed of reactions, chemical reactions within the neurons. And that changed the way that the neurons in that part of the brain were working. And so they trained these mice to click a little lever to basically to tell the researchers whether or not time was faster or slower in their head, like whether the mice were perceiving it as faster or slower. And they were able to get the mice to start telling the researchers, oh, I have a hot brain, time sped up. I have a cold brain, time slowed down for me. It took me longer. It seemed like drinking that water took forever. They're not having a conversation with the mice, but in this work, what they determined, and the really interesting part of this that they determined, is that there is this structure in the mouse striatum where the perception of the time is changed, but it doesn't actually change the movement or the motor control of the mice to actually click that lever or do the work that they're doing. And so the question is, is this part of the brain now independently dictating this just brain perception of time? And there's another area of the brain that might actually change the speed of the behavior. And they think the cerebellum is responsible for changing the speed of the behavior that happens as well. So even though the mice were like, whoa, time is speeding up, they didn't move faster. So the next question is, if they go into the cerebellum and heat it up or cool it down, will they be able to match this time perception with the speed of the actions of the mice? Yeah. So anyway, yeah, our neurons are responsible for how we perceive time, right? It's all clicking along with the speed of those neural interactions and what goes on there. But then everything is a dance, right? Everything has to happen in concert. It has to happen in a coordinated way. And it's just interesting to see which parts of the brain might be responsible for these things. And if we find out how the cerebellum's involved, what specifically the striatum's doing, if this is how it is in humans as well, which is likely, can this help us with movement disorders and possible treatments for people who have these, not just time perception, but things related to muscular disorders and things that are, shaking and palsies and other things that inhibit or impair action.
Cool.
Yeah.
And I don't know. If I want time to slow down, I can put an ice pack on my head, right? Is that what that means?
Yeah. So this is like, I thought this was so weird, the cooling down or the heating up of the neurons. And they had to be so specific in how much they did this. So this is inside the brain. It's not the surface of the brain, but I don't know, as things get hotter and as our climate gets hotter and people are boiling on the inside, what's that going to do? But anyway,
go faster.
Yeah, we got to go faster towards our demise. But it's, the specificity of this research, I think the technique is fascinating also, using the optogenetics to very, very specifically alter the internal temperature of the brain so that different functions happen. And nobody's done that previously. So this could also be used for other research questions as well. But yeah, don't just put an ice pad on your head. Don't put a bag of peas on your forehead. Don't put your head next to a heater. No, that's not what I'm saying.
Sure. Sure, that's not what you're saying, Kiki. I get it.
It's not what I'm saying.
No, I know. So okay, I have one question about this though.
Okay.
So how do they? I want a little more explanation of how they know how the mice are perceiving time. Is it at a certain point they were supposed to do something for a treat or something? So it's like once an hour you get a treat, and so it was a question of when they go to do that.
Yeah, so they trained the mice to report whether the interval between two tones was shorter or longer than a second and a half. So 2 tones would be always at a second and a half, or during the training, they were probably closer together or farther apart to train the mice to respond in a particular way.
Okay.
And they found that when they cooled the striatum, the mice were more likely to say an interval was short. When they warmed it, the mice were more likely to say that it's long.
So it was the opposite. So, okay.
So heating up the striatum sped up the neurons in the striatum, accelerating the clock. So the judgment of the time was that it was longer than it was.
Longer. Okay.
Yeah.
So I got that backwards because speeding up means speeding up time. No. The perception of time sped up. Therefore, time felt like it was taking longer.
Yes.
So it's like your Quicksilver from the X-Men where everything's moving slow around you. Got it.
Yes.
I'm there. I've arrived. Okay.
Yes.
I get it. That's a very cool experiment design.
Yes, it is.
That's pretty creative. I like it.
Yeah, and I mean, it is invasive in that, in optogenetics, you often have to have the light going into the head of the animal. So this isn't free running, but they were really able to, influence this control without having to do the genetic modification, having to change the genes. It just changed the speed of how the neurons were acting, whether they went.
I mean, if you want to make a time omelet, you gotta crack some mouse skulls, I guess. As the age-old saying goes.
As it goes. Oh, wow. I'm never going to look at omelets the same again.
Yeah.
Timey-wimey, that's for sure. Do we have any more stories?
No, that's all of it.
We cracked all our mouse skulls. Okay. Oh, my goodness. Thanks, everybody, for listening. I hope that we have challenged your heads and brought you new information today as well. And I hope that you enjoyed the show. Oh my gosh, it's time for some shout outs. Vada, thank you so much for your help on show notes and social media. Gord, Arnlore, thank you for being in the chat rooms and helping to keep things friendly. Identity 4, thank you for recording the show. Rachel, thank you for editing the show. Everyone in the chat rooms, thank you for chatting and being here while we're doing the show. Love having you here so that we can see what you're saying while we're chatting ourselves. And of course, I do want to say thank you to our Patreon sponsors. Thank you to Craig Potts, Mary Gertz, Teresa Smith, Richard Badge, Kent Northcote, Rick Loveman, George Corris, Pierre Velazarb, John Ratnaswamy, Carl Kornfeld, Chris Wozniak, Vegard Shefstad, Hal Snyder, Donathan Stiles, a.k.a. Don Stylo, Ali Coffin, Reagan, Don Mundus, P.I.G., Stephen Alberon, Daryl Myshak, Stu Pollock, Andrew Swanson, Fred S104, Sky Luke, Paul Ronovich, Kevin Reardon, Noodles, Jack, Brian Carrington, David E. Youngblood, Sean Clarence Lamb, John McKee, Greg Riley, Mark Hessenflow, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Richard, Brendan Minish, Johnny Gridley, Remy Day, G. Burton Lattimore, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, John Atwood, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Rick Ramus, Philip Shane, Kurt Larson, Craig Landon, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Kevin Mitt, Adam Mishkon, Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul D. Disney, David Simmerly, Patrick Pecoraro, and Tony Steele. Thank you all for supporting us on Patreon. And if you are interested in helping out over on Patreon, head over to twist.org and click on that Patreon link on next week's show.
We'll be back on Wednesday at 8 p.m. Pacific time, broadcasting live from our YouTube and Facebook channels, hopefully Facebook, and from twist.org/live.
Want to listen to us as a podcast? Look for This Week in Science wherever podcasts are found. And if you enjoyed the show, of course, get your friends to subscribe too.
For more information on anything you've heard here today, show notes and links to stories will be available on our website. That's at www.twis.org. That's TWIS.org. And you can also sign up for a newsletter that may sometime come. It's like waiting for Godot. Who knows?
You can contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or Blair at blairbaz@twist.org. Don't forget to put TWIS into the subject line so your email doesn't get spam filtered into a music festival where all the bats are running away and the music's too loud anyway and we just can't hear it.
Yeah, that's
No good.
that's a surefire way to be ignored.
And
You can
we will
also
avert our eyes.
Yeah, you can try to hit us up on Twitter. I saw someone tweeted at me today. My Twitter's broken. I couldn't get to it. But you can certainly try. That's at TWIS Science, at Dr. Kiki, at Jackson Fly, and at Blair's Menagerie. We do love your feedback. I would suggest sticking to that email stuff from before. If there's a topic you would like us to cover or address, or a suggestion for an interview, please let us know.
And if you learned anything from the show, wait, what am I doing? Oh, yeah, no. We'll be back here next week. And we hope you'll join us again for more great science news.
And if you've learned anything from the show tonight, remember,
it's all in your head.
This week in science. This week in science. This week in science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer and it shouldn't be news. That what I say may not represent your views, but I've done the calculations and I've got a plan.
It's the after show.
yet understand,
You may just yet understand. I've had Neil Shirley, who was one of the people involved in writing that song and producing that song, over on Threads, saying hi recently. He sent
That's
a
fun.
fun picture of some wind chimes with a sign in front of them that said that the wind chimes were made out of discarded robot parts and that they would be used as a warning to other robots.
Good. Good.
Right.
Good.
So I need to get those for, world robot domination one day.
I don't know. I don't know if you want to anger the robots, Kiki. I kind of
No,
think you got to befriend them at this point.
It's true. I've always said I am a friend of the robots. I am a friend of the robots.
I may be scared of them, but I am not going to disrespect. It's a healthy fear and respect. That's what I have.
Healthy. It's good to be a little fearful. I'm afraid of people too. What are we capable of? What's going on out there? We got to watch what we're doing.
Who made the robots? People. So
there are some researchers at Carnegie Mellon who are creating robots for exploration purposes. So like they can be controlled remotely, or they can kind of be like Mars rovers, where it's kind of a mix of both, where they got a little remote control, a little programming, or they can be completely autonomous. And I guess the thing about them is that they're like durable and able to really go explore. Like it could go into caves or places that have had earthquakes, or, I don't know, maybe they could even go out into space. But it was funny. I was like reading the story. It's like, oh, that's big advancement. And I'm sure it's great for this particular use. But I'm like, I already have a Roomba. My Roomba goes and maps my space all the time. It's like, you put a chair there? Okay, great. We already have exploring
Roomba will
robots.
remember this.
We have exploring robots in our houses. How's it going this week? You good?
Pretty good. Chugging along. Waddling along. How about you?
I'm good.
Good.
I'm tired.
Yeah. Tired. Hot.
Hot. It's warm. How's this heat wave going for everybody?
Shoosh.
Shoosh?
Shoosh.
How's the Bay Area?
It's okay. It's pretty hot. It's been in the 90s in San Jose where I work. It's been in the like 70s where I live mostly, except
that's nice.
There have been peaks like once a week for the last several weeks where it's getting into the 90s, and it's like, I don't love this. I don't have air conditioning. But it's fine. It's, I have breeze coming off the bay. I'm pretty comfy.
Eric Knapp from Alaska says, what heat wave?
Yeah. It's those drips coming off the ice on your porch. That's the heat wave.
Yeah, it's different everywhere. Like I was reading something recently and I smacked my face because it's one of those head desk situations, because it's just like, really, like finally climate scientists are over, can't even believe how fast things are changing and what's going on, and this is whatever. I'm like, are you kidding? Climate scientists have been talking about this for
they've
decades.
been knowing. Yeah,
they've been knowing.
I heard
other people haven't been talking about it.
Arizona this week, it's been in like the 110s, and the low at night, which is the thing that really gets you, the low has been like 95 to 100 depending where you're at in Arizona, which that's the problem, is if it's not dropping, then your body can't regulate, like your body can't recover while you're sleeping. You're just sitting there sweating out every drip of liquid in your body all night. That's really dangerous. Not to mention, if the grid goes down, then you're really screwed.
Yeah. Other places are, so it's super hot. Death Valley has been experiencing some incredible heat recently. Apparently there's like extreme heat tourism now. Like,
what?
People
that sounds terrible.
Yeah, there are people who are like traveling to Death Valley to take pictures next to their digital thermometer of
no thank you
how hot it is.
Currently in Death Valley right now, guess what the temperature is?
Too hot. What is it?
107.
Oh my goodness.
And
10 o'clock at night.
Yeah. And the high today was 118. On Friday and Saturday, the high is going to be 124.
Y'all. It's amazing. I mean, it is Death Valley, but still. Death Valley
on
needs
Sunday, temperatures in
also.
Furnace Creek reached 128 degrees.
I never want to live anywhere named Furnace Creek.
No.
I'm sorry.
Oh my gosh.
Let's see. Midwinter, and is it Pamela? Midwinter, and I'm running my air conditioner to bring the temperature down to 26 degrees Celsius, 78 degrees Fahrenheit. Wow. That's And Eric Knapp, meanwhile, is the coldest summer in the last 15 years, which maybe because of how the jet stream works and how the heat is building up along the southwest right now. Maybe that is pushing the colder air up. So keeping it
Could be.
cold up north.
It's all a system.
It is all a system. It's a beautiful, wonderful, complicated system that we keep, inserting ourselves into.
Well, I don't live in Arizona and I don't live in Death Valley. So I'm choosing not to insert myself into those systems.
Same. Yeah. I'm really thinking a lot recently about like just the conversations generally and how things are shifting. There's still people moving to Arizona.
Yeah.
Which I don't understand, but
yeah.
But there are more and more
people. Choice.
Yeah, but then there are people leaving Arizona and moving other places that are going to be more temperate.
Well, and it just sucks because, like, California and often Oregon covered in fires. So
yeah, we had a little bit of smoke today. Like, it's just the beginning.
Yeah, so there's no safe bastion from climate change. That's the thing. You have to
adapt, survive. It's what we must do.
Mm-hmm.
Yeah, in the torrential downpours, there's been lots of rain. New England, Vermont, Virginia, massive rain. It's just what's happening right now. The new normal.
Yep.
Yikes.
Oh
my goodness. You're moving to the desert where it's cooler. That doesn't make sense, but it does. I guess because of it'll be a dry heat instead of the humid heat.
Yeah, and the desert's colder at night.
Usually.
Like the temperature differential, I think, between day and night in the desert is usually wider.
It's not Death Valley.
If it's not Death Valley, you're right. Even so, that was an almost 20-degree difference even now.
That's true. Yeah. But yeah, I that can't be. Is that normal for this time of year? Is it getting a smaller differential? Is that what's happening?
Looking at historical temperatures for Death Valley.
Ooh. Yes, you can look them up instead of me just asking you questions. Blair, you know these answers.
I can. The internet can provide. Summary. How do I read this?
Yeah, Paul Disney. Also, no pollen or less pollen in the desert. That's for sure.
What am I looking at? I don't understand this. Let me try a different site.
What is this? What's going on?
The data is very hard to see. Okay. How about this?
Once we all live in space. There's no pollen in space, that's for sure. Potato dust.
It's a mystery. We'll never know.
Somebody has this data.
Oh, National Weather Service. How about this?
There's more and more people in Antarctica. Who?
I'm looking at Death Valley temperatures in July. The record high max. Why? Oh, the date. Got it. So today, for example, is the 19th. The record high was 128 in 2009. And that was so 128 The record low is 96. And the average, the normal, is 118 and 92.
Okay.
So that's, you know, that's a
It's not. It's getting hotter, but the nighttime temperature is
20, what is that, 26
Yeah.
degree difference? Yeah.
Yeah.
But the record high of 128 and the record low, what do you think? Oh, what's a record low maximum and a record low minimum? Oh, I get it. So
So it's the hottest or the coldest it's ever been. But
Right.
yeah, I was thinking
So-
of more like the average spread, whether it's like usually like 20 degrees different or whether it normally cools down more at night. But it
yeah,
sounds
so
like it.
118 and 92 is the average. Yeah.
Yeah.
So okay, yeah. So record low maximum. So the coolest high for the day is 96.
Huh.
And then the record low minimum, so the coolest cool for the day was 75.
Okay.
And the record high minimum was 102, which it's currently 107. So if it dips down more,
Yeah.
it might not break a record, but it might break a record
No.
this year. That's really interesting. You know, go
Yay,
fig.
record breaking.
Weather.gov had me figured out. And let me tell you, it was just for fun, I'm going to tell you, it was the 1, 2, 3, 5, 6. It was the 6th result on Google was weather.gov.
I've been having conversations with people recently about the Google browser not giving great results anymore. Like they have switched things around. It's not working the way it used to.
So I feel like a search engine has a responsibility to not just give me the information I want, but to give me the most accurate information. It's not supposed to be an echo chamber like social media, right? Like if I search Death Valley historic high, weather.gov should be the very first thing I get because it is the most accurate and unbiased information. Regardless of whether like my previous browsing history indicates I might enjoy that site.
Yeah, we've got news, a bunch of news things, but not actually answering that question. And then a bunch of things people also ask. And then National Park Service finally in the midst. And then a whole bunch of video things. And then some other results that are, a mixed bag.
Right.
But it's,
Yeah, Paul Disney, you're right. A
Very
search engine's responsibility
news-focused.
is to serve me ads. I understand that. It's
Yeah.
back to this whole insurance conversation we were having before. People need information just like they need health insurance. Like it is a public service, but it has been turned into a capitalistic nightmare. And so the bottom line. Got it.
Yup. Yes, a VPN could help, I think, but I don't know. People use other browsers. I don't know. There's other
but wouldn't a VPN, you'd still get the things
content for
that is
location.
paid, right? You'd still get the things that pay Google to be at the top of the site.
Yeah. Unless you have like ad blockers and there's all sorts of things. DuckDuckGo, isn't that the one?
Yes.
No ads. Firefox, supposed to be open source and happy
Firefox
friendly.
Does it even still exist? Good Lord.
Yes, it does. But yeah, I know. Oh yeah.
But Firefox is a browser. It's not a search engine, right?
Oh.
And DuckDuckGo is a browser. Oh no.
Yeah.
Yeah, DuckDuckGo is a browser.
Engine.
It's not a search
engine.
But
they have their own search engines.
They do?
Yeah. I think. Pretty sure. Yeah. Different search engines. I don't know. It's all sorts of stuff. Google is not the only search engine, everyone. Just putting it out there.
I would
Ooh,
rather
Sunny
die
Knox.
than use Bing, so
Yeah, DuckDuck is a UI for Bing, according to Sunny Knox.
there you go. Bing's terrible. I'm so sorry. I hope no one from Microsoft is listening or is upset about it, but Bing is the worst. It's so bad. It's like it's even worse than what I'm complaining about Google doing. It just gives you nonsense.
Oh, now it's like it's like AI powered. Didn't they do
Right.
that? But yeah.
Yeah.
So is Google. Yeah, ask Jeeves. Oh my goodness, Paul Disney.
I'll ask him, all right.
Oh, that's good. You know what works also? The library.
Oh yeah, that thing.
Librarians are amazing. They know so much. They
They're
can
so
help
good.
you so much. And libraries are
Libraries.
fantastic. I love libraries. Seriously. The more things change, the more I really just want to become a Luddite. But I enjoy doing things like this show. So, oh, people and friends. You ready for bed, Blair?
I'm so ready. Are you ready for bed?
I am so ready.
Yes. Say goodnight, Kiki.
Good night, Kiki. Say goodnight, Blair.
Good night, Blair.
Ah.
Good night, listeners, viewers, minions, friends, chatters. Good night.
Stay curious, stay safe, hopefully stay healthy. Stay lucky. We'll see you next week.